The inner mitochondrial membrane is essential for the mitochondrial function and cellular activity. Background. The inner mitochondrial membrane acts to compartmentalize the mitochondria. It acts as a physical barrier between the matrix and the intermembrane space, allowing for cellular fluid composition to differ in the two regions. This is essential for the functioning of the mitochondria. It is also impermeable for most ions and small molecules due to lack of porine proteins. This fundamentally allows for disparities between the chemical environments of the matrix and the intermembrane space, allowing membrane protein complexes to function. The inner membrane is dynamic. Crystal folds increase or decrease depending on the cell's requirement for ATP. This is mirrored by the number of mitochondria varying depending on cell type. The integrity of the inner mitochondrial membrane is essential for cell viability. If the membrane is lysed, the mitochondrial matrix, intermembrane space, and cell cytosol would have similar compositions. This would result in reduced ATP synthase efficiency, which could lead to cell apoptosis. Structure. There are two morphologically and physiologically distinct subdomains of the inner mitochondrial membrane, the inner boundary membrane and the cristae. Subdomains are linked together by a Minos complex. Minos. The mitochondrial inner membrane organization system is a large protein complex that maintains the crystal junctions between the inner boundary membrane and the cristae membranes. The complex is formed of two core proteins, mitophilin and myotin, which are essential for the linkage of the two inner membrane subdomains. Minos mutations. Mutations to the mitophilin gene disrupts the Minos complex and leads to a release of cristae membranes from the inner boundary membrane. The inner boundary membrane. This membrane is parallel to the outer membrane and is considered a second envelope. The membrane forms morphological contact points with the outer membrane, where TIM and TOM proteins are there for protein import. The lipid-to-protein ratio is similar to that of the plasma membrane, which is 1:1. TIM and TOM complexes. TIM, or transporter inner mitochondrial membrane complex, on the inner boundary membrane associate with TOM, the transporter outer mitochondrial membrane, for protein transport, are involved in localization of proteins. Membrane complexes are acquired as the inner membrane is charged. In order for the protein synthesized in the cytosol to enter the mitochondria, they require a positive pre-sequence. Once the protein has passed through the TOM complex, HSP70 binds to the unfolded protein to translocate the protein into HSP60, where the protein folds. This system requires a net loss of 3 ATP molecules. The cristae membrane. Tubular cristae increase the surface area of the inner mitochondrial membrane. Protein levels are higher, around 70 parts to 30 part lipid, as the membrane is associated with electron transport and ATP production. The intermembrane space. The intermembrane space is chemically distinct from the matrix with a lower pH and positive charge. The ion and small molecule composition is similar to that of the cytosol due to porine molecules in the outer membrane. Any deviance caused by proton pumping is isolated to the inner membrane. Protons are then free to passively diffuse out of the outer membrane into the cytosol. Protein incorporation. There are many ways of incorporating proteins into the intermembrane space. Firstly, the protein can fold once it has passed through the TOM complex. The protein can also partially enter the TIM complex and be cleaved, leaving the remaining protein free in the intermembrane space. Finally, the protein can pass through both complexes and be actively transported back into the intermembrane space through a transporter protein. ATP production. The main purpose of the mitochondria is the production of ATP via oxidative phosphorylation. This occurs in the crystal membranes as the chemiosmotic gradient builds due to proton pumping. The membrane has many properties such as impermeability, large surface area, and high protein content to aid ATP production through ATP synthase.